Welcome to this explainer. Let's just jump right in, because today we are profiling a literal microscopic killer, Corynebacterium diphtheriae. If you've ever wondered how a seemingly simple organism can completely hold the human body hostage, you are definitely in the right place. We're going to break down its anatomy, its strategy, and exactly how we fight back.
To tackle this case, we've got a quick agenda today. We're going to meet the pathogen, track the infection process, spot the clinical signs and symptoms, look at the laboratory diagnosis, and finally, cover defense and prophylaxis.
All right, part 1, meet the pathogen.
So, on the surface, Corynebacterium diphtheriae doesn't exactly look like a criminal mastermind. It's defined as a gram-positive, non-motile, rod-shaped bacterium. Basically, a thick-walled little rod that can't even move on its own. But do not let that basic description fool you. What makes this bug truly terrifying isn't its shape, it's the incredibly specific virulence factors it's packing.
This thing is armed to the teeth. Its main weapon? The Diphtheria toxin. It's a heat-label exotoxin, meaning high temperatures can destroy it, but inside the human body, it is devastating. It literally halts cellular protein synthesis, just completely shuts it down. And the craziest part? Its release is highly strategic. The bacterium actually senses its environment, and when iron levels drop, boom! That low iron is the exact trigger that tells it to unleash the toxin.
But here is the ultimate plot twist. We are looking at a massive biological conspiracy here. The bacterium doesn't naturally possess the genetic code for this toxin. It actually gets the blueprint for this deadly weapon, the tox gene, from a viral accomplice called a betaphage. It's a fascinating example of horizontal gene transfer. A virus effectively arms a bacterium to be deadly.
Moving on to part 2, the infection process.
So how does it execute its attack?
Well, the invasion begins incredibly stealthily. Step 1, the bacteria sneak into the upper respiratory tract, usually hitching a ride on airborne droplets. Step 2, they quickly latch on to the mucosal cells lining the throats. And then, step 3, once they're locked in and sense those low iron levels, they start churning out that stolen diphtheria toxin directly into the local tissue.
The fallout is immediate and severe. First, you get massive localized cell death and inflammation. As those cells die off, they mix with bacterial colonies and fibrin, which is a blood-clotting protein, to create step 2, the development of a pseudomembrane. This is this thick, grayish, horrifying barricade of dead tissue that basically cements itself to the throat, which inevitably leads to step 3, a physical obstruction of the airway. The patient is literally choked by their own dying cells.
And unfortunately, that localized respiratory nightmare, it's really just the staging ground for a much bigger, systemic emergency.
The real crisis hits when that toxin breaches the bloodstream. Once it's in systemic circulation, the clinical aftermath violently expands. The toxin launches secondary, highly lethal attacks all over the body. We're talking myocarditis, which is severe inflammation of the heart muscle. We're talking polyneuritis, causing debilitating nerve damage, and it goes right after the kidneys, too. The local infection turns into a full-body assault.
Which brings us directly to part 3, clinical signs and symptoms.
If you are a medical professional, one of the most visually alarming red flags of a severe respiratory case is massive cervical lymph node swelling, along with soft tissue swelling in the neck. It gives the patient this highly distinct bulging appearance, medically known as bull neck. When you see that, you know for sure the disease is deeply entrenched and highly toxic.
But we do have to contrast this respiratory nightmare with its milder variant, cutaneous diphtheria. Let's compare the two. While the respiratory version is severe, giving you the bull neck, pharyngitis, and that airway-blocking pseudomembrane, the cutaneous variant is a completely different clinical picture. It's less common, usually pops up in warmer climates, and shows up as chronic grayish ulcers that just won't heal. It's bad, definitely, but thankfully it results in far less systemic toxicity.
All right, part 4, laboratory diagnosis. Let's see how the medical detectives actually catch this thing. It starts with a throat or wound swab, followed by immediate microscopy. And under the scope, the forensic evidence is honestly kind of artistic. C. diphtheria are these club-shaped gram-positive rods, but it's how they arrange themselves that gives them away. They form these sharp, angular visual patterns that scientists describe as resembling Chinese letters or palisades.
To lock in the visual ID, scientists force the suspect to reveal itself by growing it on highly specialized culture media. First up is Loeffler's serum medium, which specifically highlights these internal structures called metachromatic granules. Then, they use cysteine tellurite blood agar, or CTBA. And on CTBA, there's absolutely no hiding. The bacteria show up as stark, unmistakable black colonies.
But just finding the suspect isn't enough. We have to prove it's armed. To close the case, biochemical tests are required. Medical teams use the ELEC test to check for active toxin production, or they pull out the big guns with PCR, polymerase chain reaction, to definitively spot the presence of that deadly virus-supplied tox gene blueprint we talked about earlier.
Which leads us to our final section, Part 5, Defense and Prophylaxis. Once the threat is confirmed, containment is everything. The post-exposure defense strategy is a strict, three-pronged approach. First, you immediately boost immunity for close contacts with the DTaP or Tdap vaccine. Second, you launch a heavy counterattack with antibiotics, specifically erythromycin or penicillin, for about 7 to 10 days. And third, strict isolation and monitoring of anyone exposed.
And the reason these protocols have to be enacted with lightning speed comes down to a shockingly small window. The incubation period for this devastating disease is literally just 2 to 5 days. You absolutely do not have the luxury of time. Strictly sticking to these containment protocols, the vaccines, the antibiotics, the isolation, that is the absolute only way to break the chain of infection. It's a textbook, incredibly effective public health response to a severe biological threat.
Which leaves us with a pretty fascinating thought to wrap up today's explainer. We learned earlier that a viral accomplice, the beta-phage, is what actually arms this bacterium with the toxin gene. Well, as we continue to master the anatomy of microscopic villains like this, could we eventually engineer that very same beta-phage to be our greatest tool to safely disarm it? It's an incredible frontier in microbiology. Thank you so much for joining me for this explainer. Keep asking those big questions, and I'll catch you next time.